Good afternoon. Are you listening to me? Okay. Good afternoon. So we have another seminar, our special session in epidemiology and disease ecology. And today it's my pleasure to introduce Professor Pamela Patricia Martinez. Pamela has a bachelor degree in biochemistry from Universidad de Chile, and then a PhD in ecology and evolution from University of Chicago. She did her PhD with Mercedes Pascual. That was the person that opened our program. So that's an amazing link. And then she did a postdoc. She was a postdoc researcher at Harvard University, also in the US. Currently, Pamela is an assistant professor at the University of Illinois, and she's in both microbiology and statistics department. In her research, Pamela is interested in ecology and evolution of infectious diseases, and she aims to understand how climatic and demographic factors shape the population dynamics of infectious diseases in space and time, and how pathogen diversity interact and interfere with public health interventions. To attack her questions and goals, Pamela uses mathematical and computational tools, applying them to different types of time series data. So thank you so much for accepting our invitation, Pamela, and help me to welcome her.
Thank you, everyone, and thank you, Flavia, for this invitation. I'm very excited to talk, especially since it's in South America. I feel very represented somehow by all of you. And I wanted to start maybe giving a little bit of background on my own interest. And as Flavia said, I had my undergraduate degree in biochemistry, but early on during my biochemistry course, I realized that I really like math and that I was very bad at like wet lab experiments in the bench. So I started early on working with Pablo Marquet, who was an ecologist in Chile, and he kind of showed me the path of like quantitative biology, especially in the context of ecology and evolution. And because my background was in biochemistry and microbiology, I thought that studying pathogens was a very natural course of transition from a more molecular field to like a population level quantitative field. And since my PhD, I have been using mathematical and statistical tools to understand population of pathogens. And well, now with COVID, I feel like everybody is aware of how important it is to study pathogens. But in the last few years, there has been a lot of improvement and advances in the computational field, like how powerful computers are, access to human movement, health records, sequencing, and also we have been experiencing more and more natural disasters that ultimately will impact the way and the intensity of these pathogens transmission. So I divided the talk into two main kind of fields. One is like how climate factors impact the dynamics of these diseases. Like I am going to be focusing on human diseases, and then how strain diversity interfere and play a role in these population dynamics, especially in the context of interventions such as vaccination. So for the climate factors, I did a lot of my research during my PhD on rotavirus. Rotavirus is a virus that causes diarrheal disease, and it is still responsible of a large number of deaths worldwide, but mostly in countries in Africa and Southeast Asia. Rotavirus is mainly transmitted by person-to-person contact via the fecal-oral route, but it also can be transmitted through water, and that is kind of part of my own research interest. is a common cause of diarrhea among children worldwide. And as I mentioned earlier, it remains a major public health problem, especially in low-income countries. So for this particular study, I focus on incidents from Dhaka, Bangladesh. So Dhaka is the capital of Bangladesh, and it has like more than 20 million people. And the country itself has more than 160 million people. And just as a reference, the area of Bangladesh is similar to Uruguay. So it's very small in terms of area, but very dense in the sense of like the amount of people that live in this country. And it's not only highly dense, but it also suffers from the severe flood events that are triggered by seasonal rains during the monsoon. So we knew from previous work in the lab where I did my PhD that cholera is highly sensitive to these climatic variables. And not only it's sensitive, but it also shows a differential sensitivity within the region, within the city of Dhaka. These authors identified 2 different regions that respond differently. The urban core, here shown in orange, is more climate sensitive, and it seems to act to propagate the risk of infection to the rest of the city. I'm going to switch the pointer. 1 second. Oh. No. Oops, I'm sorry.
Don't worry.
I messed up with this. I don't know how to go back. Okay, yeah. There we go.
I was you just need to go in the pen, yeah.
Oh, here I found it. Okay, I'm sorry for that. I just wanted to, I think it's easier if I show this red dot. So in orange, right, we find the urban center, and then in blue, we see kind of this periphery area. So at least we work in cholera, they describe this as 2 different regions that react differently to the transmission of cholera. So even though the dogma by then was that rotavirus was mostly transmitted by person-to-person contact, we wanted to understand how flood events might have impacted the transmission of rotavirus, knowing that El Niño Southern Oscillation has had already an impact on the transmission of cholera through these flood events. So we were particularly interested in understanding climate forcing that might impact the seasonality of the disease as well as the interannual effect. So this is the data that we analyzed. We split again the city into these 2 regions, the center in dark blue and then the periphery in this teal color. And cases are shown here per 10,000 individuals. And then on the right y-axis, I'm showing the annual flooding in the percentage of the country that was kind of covered underwater. And you can see that even by eye, during 1998, which was a very bad El Niño year, there is more cases than observed in previous or after, like before or after. And then in the bottom part, there is the seasonality. So it's very interesting because the core in blue of the city shows 2 types of peak. One during winter, which is very common for rotavirus, winter being like December, January, February, and one that is almost the same size during the monsoon, during like June, July, August, September. This monsoon peak, however, is less dramatic in the periphery of the city. And the periphery shows seasonality similar to the one observed in MATLAB, which is a rural area southeast of Dhaka. So with this data, we wanted to formulate a transmission model, a mechanistic model. And I'm not sure if you are already very familiar with these models, but I will walk you through this compartmental model, and I'm happy to answer questions at the end. So we first just say, okay, when there is newborn in the population, they enter in this M class, which are newborn individuals that have some protections due to maternal antibodies. Then with this maternal antibody wanes with rate omega, and then they become fully susceptible. That will be S1. Once they are fully susceptible, they are at exposure, or like they are at risk of getting sick. And with the force of infection lambda, then they can become infected. With the recovery rate gamma, they can become susceptible again. And then we incorporated these additional parameters sigma and nu, as well as sigma and nu, to kind of incorporate a little bit the complexity of secondary infections, assuming that once you have been infected once, there is a relative susceptibility, you are less likely to be infected again. Then also that there is like an additional duration for infections in the third time, and that this relative susceptibility is even smaller after the second infections. In addition, we had this reporting rate row that will allow us to kind of sample reported cases from our mechanistic models, and then later compare those reported cases with the observed cases from the hospital. We not only did this for we not only implemented this for the entire population, we also split it into two populations, the core and the periphery, and we coupled them, assuming that there was some mobility among these two populations. So this is more like better reflected in these equations, that is the force of infection. So the force of infection lambda is a function of the transmission term beta, and then the coupling between its own location, I, and the location with the other part of the city, J. So basically, the force of infection will be a function of the transmission term times the probability of encountering an infected individual from your location, or the probability of encountering an infected individual from the other location. Now, this transmission term beta is very important for incorporating the force of the climate covariate. And we did it by fitting these kind of flexible splines that will incorporate three components. One is the seasonality. So with the data, we were able to fit kind of this black line that resembles the seasonality with peaks in the, well, this is a cartoon. This is not actually for my data, but it tells you that there is two peaks that happen throughout all the years. And then you can add on top of that a variable function that tells you how the effect of flooding now is entering into this equation. And these flooding events, when years are have years with high flood events, then will be reflected in a higher seasonality in that particular month of the year. Finally, we incorporated this environmental noise that allows us to have an stochastic model. So this is a little bit how we treated the summary of how we treated the model to compare it with our observed data. And there are two kind of important components of this model. One that is it has a measurement noise and a process noise. So the process noise, right, it's incorporated by this environmental noise here that makes every t+1 time step different in the dynamics itself. And then this measurement noise that is like, given the outcome of my ODE function, when I sampling, I can incorporate errors, and those errors can be interpreted, for example, as error that has to do with the observation process. So whether a person went or not to the hospital and whether they were recorded in the number of cases shouldn't ultimately impact the transmission of the disease, right? So that will be a measurement noise, while process noise are those type of variations that do have an impact on the dynamics of the disease. So we feed this data using POMP, which is an R package that stands for partially observed Markov process. And we compare two different models, one that incorporated this flooding effect in the transmission term and one without this flooding effect. And they are nested because we can just simply assume that this weight for this flooding effects is 0 in the one that doesn't have any effect. So we use particle filters to estimate our parameters and then maximize the likelihood, do some profiles, intervals, compare our two models using likelihood ratio test and also AIC. And this is what we got. We found that the model with flooding had a better likelihood and also had a better AIC. So AIC, it penalizes by the number of parameters, right? Like if you have a model that has more parameters, you will expect it to fit better just because it's more flexible. So AIC allows us to take into account that, like the penalty of that complexity. The other interesting component is that with this, doing this fitting, you can actually find the confidence intervals of parameter estimates that are very hard to find in the literature. So we fix the average lifespan of the host to be 50 years, but then we estimated all of these parameters. So we found, for example, that the duration of infection is about 10 days. With a confidence interval of 7 to 15 days, which is very reasonable for this pathogen. We found that the waning of maternal immunity is very fast, and it usually declines over time up to like 110 days. We found that the reporting rate, right, was about 2.3. Times 10 to minus 3. The coupling from the core was very high versus it was lower from the periphery, which, well, what this is telling us is that you can interpret it, it's a value between 0 and 1, and if it is 1, it means that it's not coupled at all. If it's 0.5, means that half of the time cases are moving to one part of the city and half of the time they are staying in the city. So for the core means that 96% of the infected individuals are actually contributing infections within that core of the city. And that the periphery, which is 87, means that there is 13% of the cases that are actually contributing to the infection in the core from the periphery. And this makes sense if we consider, for example, that the main hospital is actually in the core of the city. Then we also measure susceptibility reduction and additional duration of infection. So this is how the simulations from the model look when compared to data. So here the black lines are the data, and then the red lines are the average of simulations for both the core and the periphery. And then the shaded area are the confidence intervals. You can see that the model captures relatively well the dynamics in both the core and the periphery, and I will argue that it even captures better the dynamics in the periphery. They also capture relatively well the seasonal trend in both the core and the periphery. So before we had access to all of these parameters, right, we could actually quantify the relative impact of the force of infections in the core and the periphery. And what we found is that the core, the force of infection in the core is an order of magnitude higher for the monsoon season, for those months that also exhibit the highest interannual variation around the mean. So these peaks here basically are as high as they are in the winter and exhibit they are highly variable, which is much smaller and less variable in the periphery. So the conclusion for this was that larger values in the core region are an indicator of a higher disease risk per individual in the core. We also look at the transmission rate data, and what we found was that it was a little counterintuitive when we were first doing this analysis, that is actually much more variable in the periphery than in the core. And the way we interpreted this is that the core is more endemic. There is cases all year round throughout the entire period of 20 years of data analyzed. And it's very seasonal. It shows a peak during the monsoon, but this peak doesn't vary as much as the peak during the periphery. So you can interpret this as a more of a saturated system, right? While the periphery in a very dry year has a very small peak during the monsoon, but it's only when these monsoon events are very extreme is that when you will see a peak that is of a similar size to that observed during the winter. So after this work, we were like, well, but can we learn a little bit more about rotavirus seasonality and what is actually driving this monsoon peak versus the winter peak? So globally, we know that rotavirus exhibits significantly heterogeneity in seasonality, and that in most places in the world, they exhibit high incidence in cooler and drier seasons. However, in endemic places such as Bangladesh, there is this additional peak that occurs during the winter or monsoon. In Dhaka in particular, there is high incidence all year round, and low humidity have been associated with incidence risk. So these are the cases now disaggregated at the administrative unit level. So we had around 40 or 50, I don't remember now, the number of TANAs or administrative units for the city, but we wanted to see whether we found any association pattern between the number of cases with, for example, the latitude or longitude. We did see a gradient, like that places in the southern part of the city had more cases during the monsoon, for example, here highlighted in darker blue, compared to those areas that have more cases during the winter than in like light green. When we look at the number of cases, the variability between the cases in the winter versus the monsoon, we found also highly variability among all of these places. So for example, this one here, this administrative unit has cases all year round. So it has cases highly prevalent in the monsoon season, but also highly prevalent during the winter season. But then we saw other regions, for example, this one, that only has mostly cases during the winter season versus the monsoon. So because this data is collected in the hospital, like through a surveillance that is ongoing, they also ask patients a lot of information like demographics, socioeconomics, environmental information. So we did have access to those surveys, and we conducted a statistical analysis to see if we found anything significant that could explain this pattern. So for demography, we had age and sex. We also had information about sanitary conditions, such as the use of tube well, defecation in open spaces, treatment of drinking water, and also socioeconomic factors such as father's income and type of material of the roof. I will get a little bit into what does tube well mean. And for this, we used a generalized linear mix effect model, in which Y here it's interpreted as the log odds of a case during the monsoon for individual I in location K. And then X, right, is the set of individual level independent variables that we consider based on this list. Finally, we added this kind of noise here that is normally distributed. So here is the image of a tube well. What is a tube well? It's a long stainless steel tube or pipe that goes underneath and take underground aquifer access water, right? So this is a picture on the right. And we found that the use of tube well, was the only one that was significantly associated with the difference between the monsoon and the winter peaks. In other words, what the other way to interpret this result is that there is a 53% decrease in the odds of experiencing a monsoon case for those reporting tube well use. So basically, people that had access to this clean water during the monsoon season were less likely to show a case of rotavirus during this season. So I have shown you, right, like two pieces of evidence that may us think that enteric rotavirus transmission can be modulated by the existence of this environmental reservoir that is sensitive to the water that is in the environment. One, by looking at differential response to climate forcing, and two, by looking at the protective effect of drinking water from tube well during the monsoon season. So I'm going to now speak a little bit about strain diversity, and I will briefly talk about rotavirus. This is not a published study, but it's something that is ongoing and is something that I hope I can pursue further during my position now as a faculty, but it's related with the strain diversity. So rotavirus is a segmented virus. It has 11 segments of double-stranded RNA, and it has two proteins in the outer layer, BP4, that targets that is a target of neutralizing antibodies and defines the P types for these serotypes, and then BP7, which is this structural protein that is also target of neutralizing antibodies and defines the G types. So we also had access data to MATLAB, which I told you is like a southeast rural town around 55 kilometers from Dhaka. And I was very puzzled by this plot because they have had like data, like reported cases throughout all this year. And then in 2007 and 2009, they incorporated two vaccine campaigns with two different vaccines. So Rotatec is a pentavalent vaccine that targets G1, G2, G3, and G4, as well as P8, and then Rotarix, which only targets G1, P8, that is the most dominant serotype worldwide. And just looking at this picture, right, like it's like, the overall trend doesn't seem to be affected by the implementation of these vaccine campaigns. And in fact, this light teal shaded curve here represents like just the trend from those time series. And you see that even during these periods, it seems to be higher than previous years. Another way to analyze this data, and I apologize here for the figure, is not in very high resolution, but what we found is that during this period, there were emergence of like new serotypes that maybe were not necessarily targeted by the vaccine. And that might explain in part why the prevalence of these pathogen was still high even during vaccine. Now we know, right, with SARS-CoV-2, we know that the role of like variants and immune escape is very important. And I think like this is one of the things I would like to move forward, is try to disentangle how diversity is impacting the effect of vaccination, especially in like low and middle income countries. So here we have this hypothesis that BP7 is really targeting, is creating a strong specific immune response, while VP4 is actually acting to provide a more generalized immune response. But this work is still ongoing. I'm now going to switch gears and talk about Streptococcus pneumonia and a research project that I did during my postdoc. And again, this is also targeting, trying to understand how strain diversity may impact the implementation of like pharmaceutical interventions such as vaccination. So Streptococcus pneumonia or pneumococcus is a bacteria that is responsible for 300,000 deaths per year in children and 45 deaths per 100,000 children younger than 5 years old. So this bacteria is transmitted mainly between healthy carriers. It colonizes the human nasopharynx. It can cause pneumonia, bacteremia, meningitis, and otitis media. There are more than 90 serotypes identified. And here what I mean by serotypes are like a definition that is based solely on the antigenic properties of the polysaccharide capsid of this bacteria, and that they are a target of the pneumococcal conjugate vaccine, or PCV. And this vaccine only targets a fraction of those 90 serotypes. And there are currently 3 types of vaccines, BP7, BP10, and BP13. And by the end of 2015, 129 countries were using one type of this vaccine. So what happened, and again, this is very interesting from the eco-evolutionary perspective, is that after the introduction of this vaccine, there was a shift in the types of serotypes that we were observing in the population. So non-vaccine serotypes, or NVT, were benefited from the removal of the vaccine serotypes. And non-vaccine serotypes became more common in carriage and disease because of this gap that now the vaccine types disease offer by increasing like these non-vaccine type opportunities to invade, right? And I will use the word serotype replacement to refer to this increase in disease caused by non-vaccine type serotypes in places where the vaccine was introduced. So there were 2 pieces of evidence that kind of support what we consider later to be like an effect of like negative frequency dependent selection. One is that cross-sectional, that if you look at these 2, the first plot in the right, like that the frequencies of the strains now were not correlated. So you can see here that if you look at the 2 different locations, Massachusetts and this other location here on the y-axis, you will see that there is not really a strong association. However, the frequencies of the accessory genes are highly correlated in unrelated populations. So this is what we then came with this concept of cross-sectional correlations between unrelated populations. The second piece of evidence is that there is an stability in these accessory genes that after the vaccine was introduced in this Navajo population in the US, we found that the population bounced back to the frequencies of those accessory genes that were pre-vaccination. So here we define 3 epoch times. Epoch 1 will be pre-vaccination, epoch 2 will be just after vaccination, and then epoch 3 will be several years after post-vaccination. So what it means is that frequencies of epoch 3 are better predicted by epoch 1 than epoch 2. Okay, so let's let's talk a little bit more about what does it mean, what are accessory genes, how can this be explained with negative frequency dependent selection. So imagine now that we have 4 strains, okay? And these strains are defined based on the accessory gene content. Then we have 2 serotypes. And as I mentioned, these serotypes are only defined based on the capsule information. So in my cartoon here, strain 1 is the only one that is part of the serotype A. And then I have strain 2, 3, and 4 that are part of the serotype B. And they may or may not share some of these accessory genes. So basically, an accessory gene is more of a statistical definition that implies that there are genes present in some individuals in the population, but not in all of them. So here we have our 4 strains, right? And we can play with them. So let's imagine that I have 2 individuals of a strain 1, 1 individual of a strain 2, and so on. They also have accessory genes, right? Here represented with green, gold, and light blue colors. So if I show you this population, there are 10 individuals, right? And also, but we can also count the number of accessory genes. So strain 1 and 2 have the gold color accessory gene. So there are 3 individuals that have that particular accessory gene. There are also 4 individuals here that have the light blue, and 6 individuals, the ones from strain 1 and the ones from strain 4, that have the green accessory gene. So then the question is, what happens if I remove one of those strains? So there were 2 individuals. I target the strain 1. Now I have 8 individuals, right? And I have reduced the frequencies of accessory gene 1, 2, and 3. Well, the number 3 now, because they don't share any information with the strain 1. So then the expectation is that if these accessory genes are actually under frequency-dependent selection, is that they will try to bounce back to the frequencies that they had pre-intervention at the cost of changing the frequencies of the other strains. So what is happening now is that if you look at time 3, we were able to restore the frequencies of the accessory genes, right? I have 3 of gold, 6 of green, and 4 of the light blue. However, when we look at the frequencies of the strains, are very different at time 1 versus time 3. This can be explained again by this negative frequency dependent selection that is a type of balancing selection and gives advantage to the rare and disadvantage of the common, and therefore keeps these frequencies at intermediate levels. And examples include, for example, antigens, bacterial signs, phage-resistant crossbreedings, and others. And all of these phenomena generate long-term polymorphisms in bacteria, either stable equilibrium of the phenotype or sustained cycling. So the question of this project was, can we predict changes in pneumococcal population based on the accessory gene frequencies? So for this, we use data from Navajo population in the US. So here highlighted in the map on the right? These are all the surveillance lab and sites within this population here in blue. We had 937 carriage isolates collected between 1998 and 2012. The carriage prevalence was about 60%, and 50% of the cases involved PCV7 vaccine serotype. What was interesting though, and I think that made a simple analysis is that the introduction of the vaccine did not change carriage prevalence. So carriage means that you carry the pathogen in your body, but it's not causing any harm. So you don't have any disease development. You just keep that pneumococcus in your throat or in other parts of the body, but it's like a commensal. And we have three epoch, right? We have the pre-vaccine epoch, with 274 isolates. We have the post-vaccine epoch with 398 isolates, and then a very long-term post-vaccine epoch with 265 isolates. So this is the first analysis we did. We wanted to see the prevalence before and after the introduction of the vaccine. So with dark colors, you see the pre-vaccine epoch. And with a lighter color, you see the post-vaccine epoch. And then you see in blue, the non-vaccine types. In red, those that are specifically targeted by the vaccine. And in gold color, those that are mixed types. So here we sorted this prevalence based on the abundance pre-vaccine epoch. And what you see is that after the introduction of the vaccine, they didn't keep the same ranking, right? So, now, for example, strain number 27 was no longer the most common strain after the introduction of the vaccine. And then you see other strains like 01 or 25 and 24 being much more prevalent than what we'll have expected by this prorata, or like assuming that this ranking will be maintained after the introduction of the vaccine. So here, like we kind of represent this by looking at the prevalence change and the expectation of this null hypothesis that things will decrease proportionally to the frequencies they had pre-vaccine introduction. And with plus and minus sinuses here, here in the bottom of the second plot, you can see those that change more than expected or less than expected based on that null expectation. So what we found overall is that 13 out of the 35 strains deviated significantly from the prevalence expected under the proportional increase, right? So those that were more prevalent were no longer the more prevalent after the introduction of the vaccine. And by doing a population model that incorporates this negative frequency dependent selection, we actually were able to recover that dynamics, right? So here, what I'm showing is a simulation from an ODE system that I will describe in a little bit, but you have two different types, the vaccine type and the non-vaccine type. You have the pre-vaccine equilibrium here highlighted with this shaded area. You introduce a vaccine, and then you disturb the system, for which we will later describe as a predicted fitness. And then you have the long-term equilibrium. And what is interesting, and I hope you can kind of acknowledge, is that the order, like those that were more prevalent after the post-vaccine equilibrium, are not necessarily like this one, for example, wasn't necessarily the most prevalent strain pre-vaccine equilibrium, right? So at least the model is capturing that future, that there is a disruption in the way that now these types are being ranked in the prevalence. And here what we see is that the predicted fitness, at least from these simulations, is like it will be positive if the frequency of that particular strain increased, and it will be overall negative if the frequency or the prevalence decrease, right? So there is a consistency between what we call predicted fitness and the outcome of this disruption. So let's talk a little bit about predicted fitness. So we use the, what's the name of this equation? The, I have a blind moment. Let me think for a second. Is the replicator equation. Sorry. Yes, I got it. Yes, it's the replicator equation, right? So the idea of a replicator equation is that your population sums to 1 and you only follow the frequencies of each of your individuals. In this case, I represent different strains, so I have frequencies of different strains and the overall frequency sums to 1. And then the other feature of the replicator equation is that it has a fitness component, okay? And there is an average fitness and then a fitness that can be changed depending on your model assumptions. Okay, so here we have X is the frequencies of my strain I, then omega is the fitness of each strain I, then this is the average population fitness estimated here. And then I have that the predicted fitness is this, basically this element here. So it will be the fitness minus the average fitness. What does this mean? Is that if this is positive, if the predicted fitness is positive, then I will expect an increase in the population size of that particular strain. If the predicted fitness is negative, I will expect a decrease in the population, in the prevalence of that particular population, right? And the way that we define fitness here is by looking at a matrix K of LG that has the presence and absence of accessory gene L in a strain G. Then E will be the pre-vaccine frequency of each allele L, because we are assuming that those pre-vaccine frequencies are going to be constant over time, because we assume that they are under NFDS, negative frequency dependent selection. And then FL is the post-vaccine frequencies of each accessory gene, right? So this is the way we estimated omega, and then we can compare omega with the average population fitness. So this is what we got. We compare our predicted fitness using these equations with the observed prevalence in change. And what you see that overall, right, we were able to capture at least whether these values decrease or increase, right? So positive changes in prevalence that were positive were actually associated with positive fitness, predicted fitness, and negative changes were associated with negative predicted fitness, with the exception of 3 strains here that didn't follow that expectation. We then, right, I show you here that this is a predicted fitness, but we could also estimate the post-vaccine equilibrium. And this in the data meant like 10 years after the vaccine was introduced. And for this, we predicted the post-vaccine equilibrium using the quadratic programming that usually is used to solve optimization problems. And it allows us to find the solution that minimize K, X minus E to the 2. I'm going to skip very technical details, but I'm happy to talk about that later if you're interested. So where K, right, is our matrix again of presence, absence of accessory genes, and it has the dimensions L by G. Then X is the vector with the strain frequencies with dimension G times 1. And then E, which is a vector with accessory gene frequencies with dimensions L by 1. So this is what we did. We have our presence absence accessory gene matrix K, and we also have the pre-vaccine strain frequency vector X. So then we said, what happens if we subset K for only the non-vaccine types, right? Because we assume that the vaccine knock out everything that was vaccine type. And what we got after that in the populations were only those non-vaccine types. So then we estimated the frequency of the accessory gene the pre-vaccine accessory gene frequency using dot product with these two vector and matrix to estimate E. So we said, okay, given that we know the information of the post-vaccine frequency and our K that was just estimated removing the vaccine types, can we estimate these post-vaccine frequencies knowing my pre-vaccine accessory gene frequency and my K matrix? And for that, we use this quadratic programming. So in blue here, I'm highlighting the input and output, meaning data, and then in green, some of the analysis using the quadratic programming. And this is what we got. So the 1-1 line refers to a perfect match. So imagine if you have had predicted what you observed, all the dots will fall in the 1-1 line. It will be very suspicious for any model if your predictions fall 100% your observed data, but you still want to be as close as you can to that 1-1 line, right? So our dots are somehow close to that 1-1 line. Not all of them, but at least we were able to recover, like to include all of our data in a confidence interval that includes the 1-1 line. So I think that was a good kind of sign, a positive sign. If you look at what happens with the predicted changes, now it looks a little more messy, but still we recover this trend, which is not recover if you think that now, if you do a very null expectation and say, well, what will happen if I rank all my strains based on the pre-vaccine frequencies, I remove my vaccine types, and I assume that everything will increase proportionally to how this rank was pre-vaccine. And that expectation is very bad, right? Like we don't even recover the 1-1 line, and the confidence interval even includes the kind of the flat slope, the slope that is equal to 0. So with this, I hope I was able to show you that the predicted fitness, it allowed us to estimate, to capture well the direction of the prevalence change, and that using the quadratic programming, we were accurately able to predict the direction and the magnitude of the observed changes post-vaccine in the long term. And the conclusion, I guess, of this part is that simple mathematical models based on negative frequency dependent selection on these accessory genes can actually predict the evolution of pneumococcal strain after the introduction of vaccination. So that is kind of the summary of my talk. I gave you a few examples of how we can analyze transmission rate, transmission dynamics when we incorporate climate variables, and also how strain diversity can make a vaccine strategy very, very complicated and messy, and it's something that is not very intuitive if you are looking just at the time series, right? And with that, I would like to thank people throughout like all these years that have been collaborating in these several projects and funding, and particularly now at UIUC, that is where I am based now. So I'm happy to answer questions.
Thank you. Do we have questions, guys? We are just passing microphone here for you to listen.
Okay.
Oh, there's one there.
Hi, good afternoon. Thank you for the talk. I had two questions. The first one is regarding this last part of the presentation of strain diversity. it wasn't clear to me if after the vaccination period, the disease prevalence itself went down and it's just the strains that changed, or if like the vaccination didn't make a huge impact on disease prevalence. And the second question.
That's a great question. Mm-hmm.
And the second question, if I can already make it, is if you could elaborate a little bit on the POMP, this partially observed Markov process. I thought that was really interesting, and if you could talk a little bit more about that.
Yes, absolutely. So here I have, let me enter in full mode. So what we found, right, was that the introduction of the vaccine did not change carriage prevalence. So maybe it changed the presentation of symptoms like the disease, but not the carriage. So that was in part why we use the replicator equation, because our population, the pathogen population, stayed constant over time. And I think that's a great question, because what it's telling us, right, is that the vaccine is actually suppressing severity, but not prevalence of the pathogen in the population. So yes, we assume that the population was constant, and that's why we use the replicator equation. Now, in terms of the second question, let me see 1 second, because I think I might have an additional figure to describe a little bit POMP. Just give me 1 second. Okay. Okay. So it is a very interesting question, because what it does, right, like the system, at least the way I interpret it, is very like driven by the analogy with evolution. If we think in Darwin, like the fittest, like the survival of the fittest. So you start with a set of parameters. So here, what we mean by particles is like each particle is a set of parameters. And we start with like random generated parameters. So let's say, I don't know, I imagine I didn't know the duration of infection. I started with a duration of infection of like 60 days, and then maybe a waning of immunity of 1 year and things like that. So you can imagine now that these dots are actually like a vector with all my parameters. And I don't really know if this set of parameters are good or bad at explaining my data, right? I put these parameters into my ODE system. I made them simulate an outbreak. And then I say, or not an outbreak, like these 10 years of data. And then I compare that data, the simulated data, to the observed data. What will happen is that then you will get a fitness landscape. And that fitness landscape is a little bit reflected in this curve here, in this kind of wavy curve, right? Which means is that some parameters are going to be much better at explaining the observed data than others. And therefore, now these parameters will have a higher weight. And that is how these now kind of bigger purple dots are represented. It's saying, okay, now I know that these parameters here are better at explaining the data than this, for example. And because this is so bad at explaining my data, I'm going to remove this set of parameters for my next iteration. And it not only gives more weight to those that are good at explaining the data, but also similar to evolution, the system has a way to mutate these parameters. So, and now I add some variability, and instead of saying, for example, if 10 days, the duration of infection in which my parameter was 10 days, gave a good fit, then for the next iteration, I add some noise, and instead of being 10 days, it's going to be 11 days or 9 days. So then I add variability, and that allows me to keep exploring this kind of landscape until I ultimately optimize the system and I have the maximum likelihood estimate. So there are two loops in this process. One first loop that iterates in time because I have temporal data throughout 20 years, right? So you really want to be able to capture all my time points. But then there is a second loop that iterates to improve over and over that set of parameters. And I'm happy to, like, nowadays, nowadays there is like a lot of documentation on how to use these methods. And I implemented them like 8 years ago. It wasn't that easy, but I feel like today it's much easier. There are like tutorials online and things like that. And I can share with Flavia some websites that maybe then she can share with the rest of the class if people are interested.
Yes, I will. Thank you.
Yeah, that'd be great. Thanks.
More question? Go ahead, Ron.
Oh, okay, first, thanks for the presentation. I have a question about the accessory genes because I'm not really familiar with this terminology, and
Yes.
like although you represented this in the bacteria like part of the main genome, the functional characteristics that it gave us like phage resistance and toxins, these are generally encoded by plasmids. So I would like to understand more about it. And again, I don't know a lot about bacteria, but I don't know if coccus bacteria can exchange genetic material. And if yes, it would definitely change the dynamic that we are seeing here. So what do you think about it?
Yes, no, those are great questions. Thank you. So going to the first one, yeah, I agree that, for example, some genes that give antibiotic resistance or like help defending the bacteria to the environment, right? Like to make them survive under like harsh conditions might be coded in plasmids. Here, what we are doing is actually oversimplify that. And because plasmids, right, still are just a way to represent how the genomes are encoded inside, those plasmids are still accessory genes because they don't belong to all the individuals. I need 1 second. I need to clean my nose. So I'm going to mute on 1 second, please. Okay, I'm sorry about that. I'm very allergic. I think in our very simplified way to do this modeling, plasmids are considered as accessory genes. But there are other genes that may not be necessarily encoded in a plasmid. It can be, for example, allele variations of the same gene that belongs to all the individuals, but there are like different basically variations of that particular allele. So for this purpose, we assume that whether it's the same gene that has different versions versus a gene that is present or absent in, for example, a plasmid, we treated them all equally. So it was like a more reductionist way of dealing with these accessory genes, because at the end of the day, we only care whether they were not present in all the individuals. Then your second question was related to, whether they are able to share or not these genes. And I need one more second again. And that's a great question, because in fact was one of the questions that the reviewers asked. And what we saw is that the frequencies of these accessory genes actually are very stable. So even though they might be able to move across these different strains, right? They are stable and they don't seem to move that often among these strains. But that's a great question. And I think that it may vary, like it's not, they don't only share genes within Streptococcus pneumonia, but they also have the ability to share genes with other species of bacteria, which makes the dynamics even more complicated. But I think at the end, what we are trying to argue is that it seems to us that what is under selection are the frequencies of the genes independent of which strains have them inside, right? And therefore, it doesn't really matter how frequent they are in strain 1 or 2. But yes, those are great questions. And the paper, like we did some extra analysis in the supplement to show that they are relatively stable and that our conclusions were not really a function of that.
Okay, thanks.
There's one more question. Go ahead, Ruth.
Hi, my name is Ruth. Well, I have two questions. The first one is related with the time series about climate. My question is if it's necessary to do a pre-processing of that time series in order to use it in the model, or is there raw data?
Yeah, well, I'm not sure what you mean by pre-processing, but I can tell you here, these were like monthly data that we got from the hospital, the DACA hospital, that is a specific hospital for diarrheal diseases. So they provide us monthly data, and this is like we included into the model this raw incidence data. Okay. So we didn't really process it other than aggregating it by these two regions. And yeah, and making sure that both time series were the same length. But other than that, we didn't really process the time series data.
Okay. And the other is related with the parameter lambda, that is the transmission rate and force. Yes. Is there a result of analytical computation? Like, well, I don't know why it's defined like in this way.
What do you mean?
Well, because what I see is that it's related with the transmission, that is the beta, but the other expression is related with some computation in particular, or it's just like for convenience in the model?
No, well, I can explain again this part. So
yes, thank you.
Usually, like if you see an SIR model, like a classical SIR model, the force of infection is given by, I can annotate here. So in a classical SIR model, this is equal to beta times I over N times S. So is your susceptible population times the probability of encountering an infected individual times the transmission term. The only thing that we are doing here is to expand this part into this, because we have two populations, the core and the periphery, and that's why we have the plus sign here. So here, for example, if I is core, all these left side of the equation is for the core, and then the right side of the equation is for the periphery.
Okay.
And pi refers to the pop n here. So n is equal to pi. And then I is just the sum because we have 3 different stages.
Yeah.
And the additional term alpha is just to denote the coupling between the core and the periphery.
Okay.
So I will argue that this is still very analytical. The only reason why I expanded it was because we have 2 populations and they were coupled. But this can be really like simplified to the classical SIR model in which you have beta times IN times S.
Okay. Yeah.
Is that more clear now?
Yes, yes, absolutely. Thank you.
Okay, thank you.
Do we have more questions, guys? Is it? Okay, so I don't know. Are you seeing me, Pamela?
Yeah, let me stop here so I can. Okay.
Okay, I would like to say thank you very much and for accepting this invitation and for giving this amazing seminar to our students. Thank you so much.
So thank you all. I found that the questions were very interesting, and I can share with Flavia the link to learn a little bit about pump and fitting. I can also share an additional material that maybe can be useful, that there is a book now that has a lot of exercises and like implementation in R. So if people are really interested in this type of projects, I think nowadays there is more material that can be used to play with these type of models.
Oh yeah, if you share with me, I can spread.
Okay, perfect.
Thank you very much.
Well, thank you all. Bye bye.
Ciao, ciao.